And I'll be with you today throughout this webinar series that we're starting today, and this particular webinar today. So in this series, we'll be gradually introducing machine learning in pharmacological modeling or in healthcare modeling. And in this particular webinar, I'll be giving a brief overview and an introduction to DeepPumas and our means of bridging mechanistic modeling and machine learning to one another. The layout for this particular webinar will be a whole bunch of slides and a bit of theory and explanation in the beginning, and then we'll wrap it up with a little bit of a code demonstration to see that things are actually working. And let's see. So if I can get my slides to start working, then perfectly. Excellent. So what is DeepPumas? I guess that's the first question. So DeepPumas is our means of bridging the gap between machine learning and scientific modeling, where machine learning is fantastically good at doing data-driven model discovery, at finding complex patterns inside of data that we might have available. And machine learning can be good at handling even complex data. You can throw a lot of different kinds of data at machine learning, and it seems to work. Now, I'm painting with a broad brush here. Machine learning is a huge field, and it can do a lot of things. But typically, a lot of machine learning lacks a bit of scientific understanding, both in terms of how you define the model itself, like how you encode the mathematical model that is the machine learning model, as well as the scientific understanding that you can get out of the machine learning model. Like, you can do something, but it can be hard. And typically it requires some fairly large data. So if you're trying to learn everything from data, then you need a lot of data to work to do this. And in the healthcare space, which is where we operate at Pumas AI, then data is expensive, not only monetarily, but also in terms of patient lives. So often data comes from a patient with a disease, and you're tracking them over time and you're observing their outcomes. And so every data point might be a patient that has to live through something. So data is expensive. Scientific modeling, on the other hand, sort of the opposite there of machine learning, in that it directly needs you to encode the scientific understanding that you have about the system that you're studying. So if it's about a disease, then you encode all of the molecular interactions or all of the things that you think is important for the disease progression. So you encode that into mathematics, and that makes the model itself very data efficient. And so you don't have that many parameters left to fit or to tune in that model. And it makes the model interpretable in that I can read someone else's mathematical model or scientific model, and I can pretty much see what the underlying assumptions are that went into that model. So they're easy to read and easy to interpret what they're doing. And then it's also fairly easy to run counterfactuals, or to say that if you encoded a mechanism into the model itself, then it's quite easy to ask yourself what would happen if you break that mechanism or perturb it, or what would happen if you design a drug to target this specific thing, right? So then there's a lot of benefits to scientific modeling, but creating them, that is labor intensive. It is labor intensive in two different ways. One of them is that you need to know, like if you're studying a biological system, you need to know the biology. You need to know what is happening. You need to know, if you're studying a disease and some treatment, you need to know the drug, you need to know what it binds to, you need to know what effect that has and how that might affect your clinical outcomes. And that is not always easy. That's not always something that we know. And so it's quite easy to miss some unintuitive relationships. So something that is happening in the true underlying system that you're modeling, but you just don't know about it. And so it can be hard to encode it. And it's also hard to utilize complex data. So if you have information in the form of an image or something like that, then it might be hard to know how to integrate that into your scientific model. And so at DeepPumas, we're seeking to bridge the gap between scientific modeling and machine learning, enabling us to utilize all sorts of data that we might have available together with the domain-specific knowledge and expertise that we have to create our more scientific models. And this is all in the purpose of making as good predictions or as good estimations or as good whatever it is that you're doing with your model as possible. So DeepPumas is applicable across the whole pharmaceutical industry, all the way from lead generation through clinical research and market research, all the way to the patient bedside. So it's just a concept, this concept of mixing machine learning and data-driven processes together with scientific processes or scientific modeling. That is something that is quite widely applicable. Now, to make it a little bit more concrete, a little bit, I say, DeepPumas, the core of DeepPumas is the deep nonlinear mixed effect model. And so it is bringing together 3 different kinds of modeling. It is statistical modeling, notably mixed effect modeling, together with dynamical modeling and machine learning modeling, all sort of quite seamlessly integrated with one another. Now, some of these things or some of these combinations have already been done before. In the field of pharmacometrics, for example, we have statistical modeling together with dynamical modeling. There you have your nonlinear mixed effect models, which are quite prevalent in the like drug development and the analyzing clinical trials in the clinical pharmacology space. There we go. You also have the intersection between dynamical systems and machine learning, and here you'll find some neural ordinary differential equations and universal differential equations, and that was recently invented / published. And there's also a bunch of examples for statistical machine learning models. But now we're bringing it all together, and I think that is one of the very early instances of that. So why are we doing this then? It's not just to publish cool papers, but it is because we'd be able to integrate more classes or more kinds of information into our models. So the statistical modeling allows us to encode known hierarchies in our data. So you might know that you're looking at time series from multiple patients. Every patient has their own time series. So there's variability in the measurements, and there's variability across patients. And so we can encode those hierarchies of variability with statistical modeling. Dynamical modeling lets us encode known mechanisms. You know that like molecule A binds to molecule B, forming complex C or whatever. You know where a drug is binding. You know something, and we can easily encode that in the form of ordinary differential equations. And we also have access to a lot of data. So it can be the longitudinal data, patient outcomes over time, but it can also be other kinds of patient data, such as images or omics or something else. And so we want to be able to bring everything together in order to make the best possible predictions. Now, to get ourselves started and make this a little bit more concrete, sort of step by step, I'll start by introducing the nonlinear mixed effect model. And for many here, this might already be quite well known. Bear with me. So nonlinear mixed effect models are good. So here, say that we have a time course. So this is a time course of some concentration of a drug inside of a patient's body, where you see some lines here for the dosing, some dosing events coming in, and there's something happening to the time course. That's nice. We tend to model this with a dynamical system. So a set of ordinary differential equations. And then we can say that, well, looking at the data, it seems like we have a bit of noise in here. We can actually model that noise through an error model. So now we're creating a statistical model saying that the observations are distributed according to some distribution that depends on some latent variable of ours. But if we then look at multiple patients, data from multiple patients, you will find that there's variability that isn't like appropriately accounted for by the error model. It is a systematic difference between the different patients. And so that we model with individual parameters. So we say that every patient might have different parameters going into this dynamical system before it produces the simulated observations or whatever it is that we do. These individual parameters, they're coming from a mix of some typical values of fixed effects, where every patient has or every entity that you're modeling has the same value. So it's a single parameter that is used for everyone. You might have covariates. These might differ. These are deterministic quantities that we can gather from our patients. And you might also have, or in nonlinear mixed effect modeling, you do have random effects, which are distributions. They're modeled as statistical distributions, and they're meant to describe and capture the unexplained or residual between subject variability. So the variability across different patients that you could not account for by the covariates. Now, in order to get ourselves to deep nonlinear mixed effect models, we just need a quick primer on neural networks. So neural networks is an information processing mechanism that is loosely based on the idea of neurons. So the idea here is that you have a neuron, it gets some input from a bunch of different sources, it processes that input in a nonlinear way, and it produces an output. And you stitch these together into a network, and they become very good at processing information. So one thing that is important for us is that mathematically, many, not all, but many neural networks are just mathematical functions. They're mapping from an input space to an output space, which means that we should be able to use them anywhere where you would use a mathematical function. And we use mathematical functions all over the place when we do scientific modeling. They're also universal approximators, meaning that they can approximate any functional form if they're large enough. And the form that they end up approximating is tuned by the parameters, typically called weights and biases of the neural network. And so the functional form that you get out of it depends on the training of the neural network or the fitting of the neural network. And it also then depends on what you're fitting it towards, like how you define the target. What is the loss function that you're fitting it towards? So we can define that however we want. And so we can train neural networks to, for example, help maximize the log likelihood of some patient outcomes over time. So that means that we can really use data to try to fit functional forms or discover functional forms that help our models explain the data that we see. So going into deep nonlinear mixed effect models, now let's say that we have some covariates, for example, where you don't know how to deal with them. There might be images, or there might be just numerical covariates, normal covariates that you just don't know how they are affecting the individual parameters. So you might not have a good hypothesis of how they're doing this. Then the idea here is that we should be able to use machine learning to process these covariates in order to inform the individual parameters of the patients. So you take the covariates, and from them you see if you can glean something that is specific to that patient that helps you account for what parameters this patient ought to have, and that makes your predictions going forward better. So this is one way in which we can bridge the gap between scientific modeling and machine learning. Another way is to say that what if you don't know the entire structural model? So if you don't know all of this. So say that here in this simple pharmacokinetic model, say that we don't know the clearance term. So we can either do the classical approach of just trial and error and guess a bunch of times and see what comes out and see what model fits best. Or you can inject a piece of like a neural network to approximate the function in a data-driven manner. So this was a brief primer on the deep NLME. Now I intend to pick it apart just a little bit, break it into pieces. So starting with neural embedded dynamical systems. So neural embedded dynamical systems has its, I would say, it has its origin in the 2018 paper, Neural Ordinary Differential Equations. And in that paper, the mixing of dynamical systems and neural networks were mostly in the service of solving the neural networks. It was about using dynamical systems as a scaffold for solving like continuous depth, continuous time, recurrent neural networks. Which was really quite nice. So it's a good paper, and it led to a lot of cool derivative works. And neural ODEs are fantastic in that they are able to handle continuous time, and you can sample them anytime you want, and so it doesn't have to be solved on a grid. So they're nice. But 2 years later, there was a different sort of interpretation of a similar concept, right? In 2020, there was a publication, Universal Differential Equations for Scientific Machine Learning by Chris Rakaukas et al. Chris being one of the early members of Pumas AI. And there, the idea is, again, like we can embed neural networks in dynamical systems, but now rather than letting just treating the dynamical system as a scaffold for solving your machine learning problem, we use the machine learning to help us out in our scientific model. So say that you have a scientific model where part of it is known and some parts are not. Then we can embed neural networks or universal approximators to approximate or pick up the missing pieces. Now, we have another term that I would like to just introduce, which is then scientific machine learning. So just to disambiguate these three terms, which I think is sometimes used interchangeably. But scientific machine learning, in my mind, is a more abstract concept of mixing modeling with machine learning, or scientific modeling with machine learning, where the universal differential equation is certainly a part of that, a large part of that. But SciML is a little bit broader than that. Let's have a look at the amount of information that we're encoding in one of these models. So here we'd have this would essentially be a neural ODE, really. Sure, I've put in different names to the state variables here, but we have that all of the state variables in this dynamical system is captured by a neural network, and the neural network is taking all of the state variables in as inputs, and it produces vector outputs. And here you see that we're indexing these vectors. So the neural network really decides the change rate of the entire state space of this dynamical system. So it is a neural ordinary differential equation. And the encoded knowledge here from a scientific point of view is not much. We might encode like the initial values of our states, but primarily we're encoding the number of states. And the number of states does matter. It is a constraint. If you only have one state, then you can't get anything that is non-monotonic. If you only have two states, you can't get chaotic behavior. If you have three states, you can get chaos. So there's a bit of like nonlinear dynamics theory in there. So it is a constraint, but it's a very weak one. We do not encode much information in this one. Next one though, like next model, here again, the entirety of the model is determined by neural networks. There's nothing explicit in here, but we've encoded a lot more information. We've encoded the number of states, but we also encoded all of the dependency, the full dependency graph here, of saying that depot is representing something. This is this is a classical pharmacometric model where depot is where you inject the drug, and then it gets transferred into a central compartment where it does its thing, and it affects a response R here. And so here we're encoding that whatever is lost from depot gets transferred to the central compartment. So you lose something here, but you have an opposite signed same term in the central compartment. So we've encoded conservation. We've encoded that this only depends on depot itself, not anything else. We've encoded that the clearance or the degradation of this of the drug in the central compartment only depends on the drug in the central compartment, and we have defined that the response can depend on the response variable itself and the drug in the central compartment, not in the depot. So we've encoded quite a lot more information, even though there's no terms in here that are explicit. Now we can go further and say that we can encode explicit knowledge of some specific terms. So now we have some terms that are explicit and some terms that are data-driven. And so now we have the entire pharmacokinetics is fully defined, and the drug response then is not. And one step furthermore here, we can encode that this is an indirect response model, where I'm just not sure about the functional form, about how the responder responds to the drug. And so here we have encoded the precise position of the unknown function. We've placed a neural network in a specific place, and it is only there that it can do its thing, and we've defined exactly what input it has. And so this model has a lot of knowledge encoded into it. So universal differential equations, I would say they're conceptually pretty simple, really. Mathematically, neural networks, or many neural networks, are just mathematical functions. They should be usable anywhere. Just decide where you have something that is unknown, and decide what kind of inputs that this approximator may have. So what is reasonable that, what entities might reasonably affect the thing that you're trying to model, and then just fit everything in concert. So treat the weights and the biases of your neural networks as just parameters of this ODE system, same as the rest, and then just fit everything in concert. And so the only hard part here, and the only thing that we haven't had this for ages, is that it might be finicky to get robust training for this. That's typically not the scientist's problem anymore. That is the developer's problem. So that would be on me. Next up, we'll take this a little bit to longitudinal data. So universal differential equations are nice, but they tend to deal with a single time series. But the kind of data that we often have in the healthcare space is longitudinal. So it can be, so often it is multiple time courses from different patients, or you have a time course for every patient. You can actually have repeated observations of anything across different entities. So like a typical example is patients with time courses, but you could also have something entirely different. It can be in epidemiology, it can be the number of affected people over time stratified by countries, or it can be something else. Right? But here we'll be looking at time courses across patients. And we can find here that, like, if you look at real data, then we tend to see that we have systematic differences, even if we have similar conditions. So even if you treat the patients the same, you give them the same drug, they have the same disease, you dose them at the same times with the same amounts, they still respond differently. They still have differences in their outcomes. Now, this is something that we can deal with or treat with, like partly with covariates, but also with random effects. And so using mixed effect modeling, we can use random effects to account for this between subject variability and to characterize it. And so what are random effects? So in terms of simulation, I think this is the easiest way to start. It's fairly simple. So if we take a look at this defined model here, I think details here don't matter that much. Don't read too much into the equations. But the random effects are some parameters or some values that are coming out of a distribution. We tend to think of them as a distribution. When you want to simulate a new observation, you want to simulate a new patient, then you just sample from this distribution. So the typical values and the covariates, they're all just numbers. And the random effect, you just sample from it, and then you use the value that you got to inform your individual parameters, and then you run your dynamical system, and you sample from your error model, and then you get your simulated data out. So just sample from the prior distribution and then use this as a normal value, and it's all good. So it's simple. Random effects are conceptually simple when you're doing simulation. But the tricky part and the important part, and why we use them at all, is not really in the simulation, but rather in the fitting. So how do we fit something with a random effect? That's the harder bit. And so one thing that you might think of would be to fit a joint likelihood or a joint maximum a posteriori, where you want to find the fixed effects. Here, I'm just calling them all theta. So the set of all fixed effects theta, um, together with the random effects. And again, I'm saying all random effects, the set of all random effects is eta, given some covariates. And we want to maximize the joint likelihood. So this would be essentially saying that I want to minimize some kind of metric that deals with the residuals. So for every data point that we have available, I want my prediction to be close to that data point. And how we penalize the differences between our or the residuals, that is sort of up to our error model a bit. But essentially, it is a minimization of a prediction error that we want to do. And so this maximizes the conditional or the joint probability that the data comes from the model. So you just tune the fixed effects, tune the random effects, such that the model is as close as possible to your data. This is not typically what we do in nonlinear mixed effect modeling. What we typically do is to maximize the marginal likelihood. So the marginal likelihood, we like try to marginalize out the random effects. This is essentially an averaging. We are taking the average likelihood over all possible values of the random effects weighted by their prior distribution. And so here yeah, we have the joint likelihood, but now we're integrating over it to marginalize out the effect of the random effects. Now this really penalizes the sensitivity to the precise values of the random effects. Now we can see or reason around how that is happening here. So here I'm just plotting. Here I'm just plotting this is the like call it the joint probability or the joint likelihood, where, and we have three different patients, and here on the x-axis I have a single random effect value, and you see that, okay, so for if you want to maximize the likelihood here, what you want to do is to find the random effect value. For patient 2, you want to find this value. For patient 1, you want to find this value over here. For patient 3, you'll want to find this value over here. I hope you can see my cursor. Yes, you can. There we go. And so you have that the fitting of your thetas, the fixed effects, it sort of gives shape to these curves. And then the fitting of the random effects here, it positions you on these curves, and you want to find the maximum value, and you want the maximum value here to be as high as possible. And so this would be maximizing the joint likelihood. Then now we are multiplying by prior, and so here we just see these curves multiplied by the prior. And the important bit here is that when we're trying to maximize the marginal likelihood, this is the integrand. This is the thing that we're integrating over. And so the maximization here that we're trying to do is not to make this peak as high as possible, but the cost here, the loss function, is coming from the area under these curves. And so you can imagine that if you want to train a model so that these, like according to this metric here, the joint likelihood, then you want these curves to be as high as possible. It doesn't matter how broad they are. And that tells you something, because if they're not broad, then it means that for most values of the random effects, the result is crap. It is only if you find a very precise value of the random effects that that you get a very good fit. And so that indicates that you've overfitted. You're very, very sensitive to a patient's sort of individual value of the random effects in order to fit your data well. In the marginal likelihood, that is completely like penalized. You cannot do this. If you just by minimizing the distance between your prediction and your data, that doesn't help you that much if it's very, very sensitive to the value of the random effects. And so the marginal likelihood helps us avoid overfitting, and it helps us give all of these nice properties of the nonlinear mixed effect models that we have, so that our fits and our models, they generalize well to new and unseen data. Now, see, and in reality, the marginal likelihood is not something that we tend, like, often we can't get access to it. We don't know what this joint likelihood is, and we can't integrate over it. So it's intractable, and we tend to use some kind of approximation. And some of you might know of the Laplace approximation, or FOCE, so first order conditional estimation or first order estimation. These are all just ways of trying to approximate this integral, to approximate this marginal likelihood. You might also try to fit your models through indirect maximizations. It could be through expectation maximization, and there a common algorithm is the stochastic approximation, SAEM. And there are more in the works here that we're working on. I'm hopeful. So if we go then to modeling between subject variability, so take a look at a classical pharmacometric model. So here, this is an indirect response model. Again, details here don't matter much. I just want to use this as an example. So what you would do is that you define the structure of the model, and then at some point you need to define or figure out where are the points at which you have between subject variability. So you decide that, okay, so we're going to have between subject variability in these particular parameter. We're going to try to account for between subject variability in these parameters. And in modeling here, it's often fairly arbitrary, or like you might have some idea of where you want to account for this. But it's often a judgment call where you put random effects or not. So typically you attach the random effects to a specific parameter, and then you can account for between subject variability at very, very distinct points inside of your model. Now, what if we did not know the drug effect? Right? So now, what if I wanted to replace this term here with a neural network, because I don't know how the drug is causing or stimulating a response in my indirect response model. So I can inject a neural network, but doing so, like this is, one might think this is all I need. This is the state variable, the drug in the central compartment. That is what I want the model to be able to respond to. But in this model here, this classical model, we have a few different points at which we can account for between subject variability. And so how do we do this when we embed a neural network? So typically, or the way that we approach this at DeepPumas is to say that the parameters of the neural network, the weights and the biases of the neural network, they're just fixed effects. We don't want them to be random effects. That would be too much, way, way too much freedom. And so everyone has the same parameters for the neural network. And that sort of corresponds to the neural network, when training its parameters, its weights and biases, it assumes a functional form. And every patient has the same functional form, which is similar then to how you define a classical model. You define the model, everyone has the same structure. There are some points at which you have variability, but the structure, the equations here, there is all shared. So everyone has the same model. They just have slight differences in their parameters. And so when we embed the neural network, we want to do something similar. And first off, I would say that, okay, so it's not really central, the amount of drug in the central compartment that we want to be using here. It's rather the concentration of drug in the central compartment. And we already had a random effect attached to this volume of distribution. So now we normalize the inputs by something that does have a random effect attached to it. So now we're able to account for between subject variability in the volume of distribution, similar to what you did in this model here. But still, this whole term here, we have the S-max and the SE50. These are two more parameters that have individual variability. And so here we only have one random effect, 1 degree of freedom along which we can integrate out between subject variability. And here we had 3. So in order to rectify this, we'll just have to add some degrees of freedom. And so this is how we would do that. We simply add 2 more random effects. And now, like, it gets a little bit weird for some. Their exact definition here don't matter that much any longer. So before you'd have that the random effect on the K out here, it's supposed to mean something. It's supposed to like be attached to something that might be measurable in the biological system. Now we're sort of surrendering that a little bit. We're saying that I need 2 extra degrees of freedom in order to account for the variability that we see, the heterogeneity that we see in the outcomes of the patient. But the neural network is quite flexible and can, whatever I do to these ADAs here, if I exponentiate them, if I multiply them or add some additional values to make them represent some biological property, like the neural network can transform them at will. It doesn't really matter. Everything I do becomes linearly dependent or dependent on, like, the neural network can do nonlinear transforms of this anyway. And so it doesn't make sense for us to try to pre-process these random effects. We just add them straight in. So these are just eta 1 and eta 2 are typically standard normally distributed random effects that we add in. And then the neural network needs to figure out itself how to utilize this in order to have good effect. And so now we're coming back again to the marginal likelihood. Getting back here a few times. So the marginal likelihood, it is a multivariate integral. So it tries to integrate across this multivariate integral where we're integrating over every one of the ADAs, every one of the random effects. And so every random effect is a single dimension in this multivariate integral. And so every random effect integrates, allows us to integrate out 1 dimension of between subject variability in the outcomes in the time series of our data. And so with 3 random effects, then inside of our neural networks here, we'll have 3 degrees of freedom or 3 dimensions along which we can integrate out between subject variability. And we can also see here that one of these dimensions is much more constrained than the other one. The volume of distribution has an interpreted meaning. It is constrained, whereas these 2 are highly unconstrained. And the neural network will, like during fitting, the neural network will have to try to figure out how to utilize this. And so we can go, like here, I can easily spend a few days talking about this. And indeed, in many of our workshops, we do, but I don't have time for that today. So instead, I think we'll go straight to a, oh yeah, I should also mention next to my slide, the marginal likelihood that we use here, it penalizes overfitting. But to see this, I'd like to go to a software demonstration. So just I want to show you the DeepPumas software and how this is done. And so we can reason around this. And I should also mention that DeepPumas right now, it is, we have a cloud and a desktop application for DeepPumas. I will be using the cloud application today. And we are currently at version 0.8, which is good if you're looking at this in the future, if this is recorded. And it is a beta release, so there's no guarantees that we won't be breaking things, which is good for innovation and for progress. But at some point, we'll have to reach stability too. Now, so DeepPumas, the way that I would interact with this is through Julia Hub, one way or another, so contact sales. But in Julia Hub, I open up this web interface, see removal of my stuff here, and I can open up a DeepPumas application. And so here I can launch a new instance or launch a custom instance, and you can see I can pick like the machine type, like how much power do I need, like how many virtual CPUs, how much memory do I want to run it on a GPU. And then a time limit, how long do I want to run it, and I will start this off, and it takes a minute or two to get it running. I already have one running here to save us that time. And so once you open it up, you'll get to a new tab, which is just a VS Code tab. I should hide that. There we go. And so now this is DeepPumas. I should have cleared this. And DeepPumas is really just like, it's an application. It's a package inside of a full programming language. So we're using the Julia programming language. And in order to use DeepPumas, you start by just loading it. And so using DeepPumas, and now I have all of this bridged machine learning and scientific modeling functionality at my fingertips. Now I'll be loading other packages too, which is nice. You can utilize the Julia ecosystem of packages here. This is the plotting package, Kairamaki. I can load data frames, some Corda tools, some CSV tooling. I can set some options here for plotting. Now I'll be reading some data in. So read some data. This is a CSV file. I'll read it in as a population, Pumas populations. Now we're just formatting this data into something that we can work with in Pumas. And a population here is really a vector of different subjects. So we're taking this data frame and it has a special format where like you have an ID column and you have a time column and you have some things that demark what is a dose and what is the dosing amount and where is it dosed into and what are the observations. So we're specifying what part of our data frame here are outcomes or observations and what part might be covariates. And then you have some predefined or default values for what columns are time, what columns are the patient IDs and things like that. And then we read it into a vector of subjects. In this particular case, like here, my data set is large. I have like 1,500 subjects here. I don't need that for this presentation or for this demo. So I'm gonna take a subset of this. And this is nice. The population here is just a vector of subjects, so I can index the vector and I can slice it. So I'm going to define a training population of 12 subjects. And we have a test population as well that I won't be using for fitting of another 12 subjects. We can just have a look at them. So plot them. There we go. We'll get them up. Perfect. So these are the different subjects. And so here we see the dosing events and they're heterogeneous. So the patients are dosed a little bit differently. And you see the data coming out of that as well. Now comes the deep NLME modeling part. So now we'll be defining a model, and I'll be starting from like reading this from bottom up. So we'll be modeling some outcomes, some clinical outcome, and we'll be modeling the concentration of the drug. And this will be coming, so their model has normal distributions around some latent variable that we're, some latent dynamical variable that is coming out of this dynamical system. And here, so now we're saying that, okay, so we're going to have a kind of indirect response model, just as the one that I had in the slides, but where there's a part of it that I don't know. So I don't know how the drug is affecting the response. And so I'm going to use a neural network to try to capture that. So rather than just guessing, I'll use a neural network. And this neural network has central over the volume of distribution as an input. And you see, I call this an IN in here. First things first, the neural network, we can define this. The neural network is defined up here in the parameters. So this is it's sort of considered a fixed effect in our parameter block. So here we define all of our fixed effects. All of the parameters are just normal parameters that need to be fitted. We define the neural network. So we define it here saying that this is a multi-layer perceptron, MLP domain, where we have 3 inputs to this neural network. We have one hidden layer of 6 nodes. We have another hidden layer of 6 nodes, and then that goes to a single output node that has the identity activation function. We'll have a default activation function of tanh for the rest of these hidden layers. And then we're adding some regularization, some L2 regularization. So like a sum of square regularization. It's similar to a normally distributed prior on the parameters, on the weights and biases of the neural network. And I said here that I had 3 inputs, and here you only see 1, and it's because in this pre-block here, we're pre-processing some of our parameters to like essentially attach random effects to them, and I do the same thing for the neural network. So I take the neural network and fix here. What it's essentially doing is using an efficient way of saying that the first 2 inputs of my neural network will always be random effects. And so here eta nn is a coming from a multivariate normal. It has a single it has 2 dimensions. So this is a vector of 2 values. So I'm taking this vector of 2 values and I'm attaching it to the neural network and giving me this individualized neural network that I then provide the third and final input to, provide the third and final input to this one, which is then the concentration of drug. And then the rest is more classical NLME or pharmacometric modeling. Now I define this model, I can execute this, and then I want to fit it. Let's see, I have a very, very generous time limit here. We'll see, it'll take a few seconds before you start seeing some results, and this is because the model is being compiled. Julia is a just-in-time compiled language, so now we're compiling things, and then soon we'll see how the fit is proceeding. But we can note here that in the fit here, we define that I want to fit the model. I want to do it on this training population. This training population had 12 patients in it, so it's not a huge data set. This is not like we're training something with machine learning in it, but we're not using massive data. This is something you might have in preclinical. This is something you might have in rare diseases. So like this kind of methodology is not limited to large data sets. Then we're just taking the initial parameters and we're fitting towards the maximum a posteriori approximated by FOS. And so the map here, maximum a posteriori means that we're respecting this regularization that we had in the neural network, which was then implemented as priors on the parameters, too detailed. But importantly, like we're using FOCE. So we are fitting towards maximizing a marginal likelihood. And this is, as we've mentioned before, like it is important for generalization performance. So here I'm going to make a prediction from the training data. So this will be the data that we actually trained on. And so here, this is the results from the deep NLME model on the training data. And so here we see 2 different kinds of lines. So we have the PREDS in blue and the iPREDS in yellow, where the PREDS is just setting the random effect values to the mean of the prior distribution. And so here it's just they're all set to 0. And so here we don't have much between subject variability, and the only way in which you have differences between the different patients here is because they have different conditions on the model, so different dosing They all get the same dose amount, but they have different dosing times. And let's see, the individual predictions, on the other hand, these are not true predictions going forward. It's not something that we can do from time zero, but they're rather post hoc predictions in that we observe the data and then we estimate or calculate what were the optimal values of the random effects in order to make our model fit these particular patients as well as possible. So I tend to call this computing the empirical base estimates. And then we use these empirical based estimates as the values in the model when we make the prediction. So the two different kinds of prediction, one of them being the true prediction, which is not heavily informed in this case on patient-specific properties because we haven't encoded them. We haven't used covariates in this model. And then the yellow line is for the individual predictions. So this was for the training data. And then we'll do the same on the test data, because when you have machine learning involved, and actually I would argue in many cases in modeling, it's a good idea to split your data so that you have something that's withheld from training to give yourself an assessment of generalization performance. So now I'll be plotting this, there we go, on the test population. And we see that, again, the model does admirably even on test data. So this indicates that we have found something that seems like reasonable. It is capable, what the function that a neural network has found is capable of reproducing the exact same behavior as like the data generating process is doing. It is finding the correct relationship between the drug and the effect or the response, and it does so in a way that can be individualized, enabling us to account for the range of variability that we have across our patients. So we've been able to utilize machine learning to pick up a small piece of our model in a way that that individualizes well. And so there you go. And so just quickly here, then this doesn't have to be hard, right? So we've embedded a neural network, but the syntax here is not hard. It is not conceptually that hard. Like, it takes a little bit of mind bending to understand exactly what is happening. But in terms of just generating and creating this model and making it run, it is not particularly hard. This one fitted in a few seconds. I can rerun it here just to see. So it doesn't have to take a long time. If you have a larger model, it will start taking a long time, but like it doesn't have to be that bad. And I should also mention that we are inside of this cloud compute environment. So if I do have a model that is much more complex than this, and you can look at our tutorials, for example, in our documentation, we have some examples, then we can also use cloud compute here. So here's the, oh, there we go, my past DeepPumas jobs. I can take this file and I can submit it to a batch job, a cloud compute batch job, where I can decide like the shape of my machine, like how many nodes, how many processes do I want to distribute it, how how much do I want to use, how many processes per node, and see node type, there we go, how many processes per node, 8, and how many nodes, 1,000. And now my boss is anxious so that I don't click the submit button here. But essentially I can scale up compute pretty easily. And then just submit this and get the result back, and it's all good. So coming back to my slides here for a very quick conclusion. We in DeepPumas, we have this core technology, the deep nonlinear mixed effect modeling, enabling us to mix statistical, dynamical, and machine learning modeling. And this is in order to utilize more information. So domain-specific knowledge that we can encode into the model. We can pick up the pieces that we don't know. We can add complex data, complex covariates. And I haven't touched upon this in this webinar, but I'm leaving room for it for the next ones, because a lot of the next webinars will be about that one way or another. And we can utilize then hierarchically structured data, so longitudinal data that is prevalent in the healthcare industry. And to learn more, then we do have further webinars. We're aiming at making this monthly for the time being. Well, we'll have the next one will be about harnessing generative machine learning to enhance simulation accuracy. And this will really start getting at a different way of bridging the gap between machine learning and nonlinear mixed effect modeling and or scientific modeling, in exploring the similarities between generative AI and NLME model, and see how we can actually bridge them very, very neatly. We'll also have other opportunities for learning. We'll have a 5-day workshop in Basel in April, and there'll be most likely more workshops throughout the year that will be running for where we can go into more depth. And so with that, I thank you, and thanks for your attention. And if there are any questions in the chat here, I can have a look at that, if I can figure out how to do this. But we are, I don't see any. So we are on top of the hour. So thanks very much for attending, and I look forward to seeing you and to the next instances of this webinar series. Thanks. Bye.